My name is Khemakhem, and for my project, I'm investigating the effects of heavy metals on the expression of antioxidant enzymes in Caenorhabditis elegans as a model for humans. In today's society, heavy metal pollution is an ever-increasing issue. Technology and electronics use heavy metals, therefore there's more heavy metal waste, and heavy metals are known carcinogens, along with causing a wide slew of health effects, one of those being an increase in oxidative stress. Oxidative stress occurs when the ratio of reactive oxygen species to antioxidants is too large. Pretty simply, reactive oxygen species are generated as a byproduct of cellular metabolism, and oxidative stress is known to lead to a disruption in signaling pathways and physiological function, along with chemical modifications within organisms. Also, heavy metals are known to induce oxidative stress within organisms. The body's way of counteracting against this oxidative stress is through the use of antioxidants. And antioxidants can serve as biomarkers for oxidative stress. Therefore, measuring the levels of antioxidants in an organism at any given time can indicate the level of oxidative stress. Before delving into the research, it's important to answer the question, what are Caenorhabditis elegans? Well, C. elegans are a microscopic worm who are commonly used as model organisms due to their short life cycle, ease to cultivate, and genetic similarities to humans. And that leads us to the research question. Using C. elegans as a model for humans, what are the effects of heavy metals on the expression of antioxidant enzymes? For the methods of exposure, there's a 5-step plan. The first of which is culturing C. elegans colonies on non-exposed agar plates. Those are agar plates that don't have any heavy metals. The second step is creating solutions of nickel and cadmium at their respective concentrations, which I'll delve into further later. The third of which is creating agar plates with a mixture of OP50 E. coli and the aqueous metals. The fourth of which is placing C. elegans on those plates to be exposed to. And the fifth step is extracting RNA from those exposed and control C. elegans. For the nickel and cadmium concentrations will be 0, 1, 10, and 1,000 parts per billion. And these are based off their natural concentration in seawater, 5.44 parts per billion for nickel and 0.48 parts per billion for cadmium. And for RNA extraction, I'll follow the protocol from the Monarch Total RNA Mini Prep Kit, and after extraction, any RNA not being immediately used was stored at negative 80 degrees Celsius. That brings us to the data collection methods, the first of which is running a nano-drop quality assessment trial to get the concentrations of RNA. The second of which is programming the RT-qPCR with the correct information for proper data collection. The third step is running the RT-qPCR and collecting data on the expression of the 3 enzymes. And the fourth step is data analysis, and that's analyzing RT-qPCR data using the delta-delta CT analysis technique. For our preliminary results, the first of which being visual observations, we found that plates with higher concentrations of heavy metals had more visible death upon them. The second of which is the primer design, where 3 primers were successfully designed and ordered from IDT, Integrated DNA Technologies, and those can be found in Table 1. And the third of which is nanodrop concentrations. The RNA concentrations were tested using a nanodropper, and those can be seen in Table 2, the concentrations, and then the 260, 280 values and the 260, 230 values. For the results of the worms exposed to nickel, exposure to 1 part per billion led to an increase in catalase by 1,175.6%, glutathione peroxidase by 2,032.3%, and superoxide dismutase by 583.9%. Exposure to 10 parts per billion led to catalase increase of 181.6%, a glutathione peroxidase increase of 177.5%, but superoxide dismutase was only 93.7% of the control trial. And exposure to 1,000 parts per billion led to an increase in catalase of 341.6%, glutathione peroxidase of 373%, and superoxide dismutase of 297.8%. This is a graph of the worms exposed to 1 parts per billion and their expression fold change in relation to the control. This is a graph of the worms exposed to 10 parts per billion of nickel and their expression fold change in relation to the control. And this is a graph of the worms exposed to 1,000 parts per billion and their expression fold change in relation to the control. For the discussion, the research found that colonies of C. elegans exposed to higher concentrations of heavy metals experienced higher rates of death, and double delta CT analysis shows that nickel exposure increases expression of antioxidant enzymes in nearly all samples. Upregulation was highest in C. elegans exposed to 1 part per billion per nickel, which does seem counterintuitive. However, there is a reason for this. That reason being that RNA extraction had to take place in 2 separate parts due to some complications, in which the 1 part per billion plate was extracted 1 week before the rest, which could be a reason for the large discrepancy between the 1 part per billion plate and the rest of the plates. For the conclusion, the research found that heavy metals increase the expression of antioxidant enzymes in C. norhabditis elegans across various concentrations. That being said, there are still steps for further research, the first of which being cadmium. After finishing my nickel-exposed worms, I'll move on to exposure to cadmium. The second of which being data analysis, in which qPCR data from the cadmium worms will be analyzed. And third and lastly, I'll compare the nickel results to the cadmium results and try to find any similarities or differences to make an overarching conclusion. And these are all of my references.